Hey, welcome everybody to our Friday session of the winter school. My name is Antonio Celani from ISTP, and I will be chairing this short session of today. As always, the rules are that you are invited to type your questions in the chat, and at the end of the talk, there will be a discussion session in which your questions will be answered. For the viewers who are following us from the YouTube live stream, you can write down your questions in the chat, and I will read them out for you in the end. Okay? So with that, I will leave the floor to Andrea Rinaldo from the École Polytechnique Fédérale de Lausanne. Will deliver the first of his 3 lectures. Thank you, Andrea.
Thank you so much. Thank you so much, Antonio. And welcome everybody. Let me share my screen. I'll be mostly counting on that. And I may be a little bit abrupt in stopping at the 45th minute, but I'm confident that because I have 3 lectures, I can compensate the material should I be somewhat get carried away in the presentation, because I kind of like the subject quite a bit. So let me show you what's the rationale. The network says ecological corridors, and the subtitle reads species, populations, and pathogens. And in fact, I think that the boy from Bangladesh, about whom I'd be speculating later on in the 3rd lecture in particular, was trying to convince me that the mighty waters of the Meghna River couldn't carry a pathogen that in fact could infect him from cholera. And given that this happened like a couple of 100 meters downstream of the largest diarrheal hospital in the world, one wonders whether our limited knowledge in fact is a permanent liability on our ability to put a price tag on ecosystem services. So here I'll be advocating for a particular robustness of our capabilities of predicting what happens in a case of a particular substrate for ecological interactions, which is through the channel network. And I am told that this is not a seminar, it's supposed to be a lecture. At times I'll try to be somewhat technical, and what I can say is that whatever I'll be doing, and I gather that what Marino Gatto will be doing as well in his lectures, is taken from a book we published. It came out like this month, or maybe the end of November. And if anybody's interested, of course, details and much details, but maybe too much details contained therein. And it had not by chance the picture which I took in Bangladesh during field work, in fact, is a very willing example. So river networks as ecological corridors. This picture is taken in my beloved hometown. It's surrounding the city of Venice. These are tidal networks, not river networks. Just for those of you more versed in geomorphology would have spotted that immediately. But anyway, the coexistence of the built and the natural environment suggests that this is a long-standing issue that has been exploited with a limited understanding for a long, long time. And in fact, my pitch will be that essentially dendritic substrates for ecological interaction, in particular that of river networks, of course, of which this is a particular, if you please, geomorphological relevant exception, but it's so beautiful, the picture, that I couldn't resist, in fact, even showing those white spots, which are nets, fishermen's nets, in fact. But anyways, my pitch is for dendritic structures for ecological interaction that bear rather fundamental consequences on a number of processes, in particular patterns of biodiversity, but much more, in my view, to control the spread of, say, a number of things with species, okay, populations, like dynamics of populations, into the fluvial connections, and from there into models of infections, like COVID-19, as Marino would be doing. So the plan of my first 45 minutes is why, in fact, I believe that we can move from rather abstract models of dendritic ecosystem, yet with some important constraints. And why they're important is the quantitative evaluation of ecosystem services. In particular, I'll be trying to show you progressively through abstract theoretical work, more refined theoretical work, and laboratory and field work that we carried out in my lab in Lausanne, that directional dispersal and the spatial ecology kind of concert the spatial situation for on an essential bearing on form and function of a river network. That is fluvial ecosystems in a sense, but as we'll try to show, much more than that. But the issue would be, again, the dynamics of fluvial ecosystem services, not today, biological invasions, not today, populations migrations, not today, and then transmission of waterborne disease. Today we'll set the premise and for these to be verified on practical cases. And again, if I may choose a punchline, I'm a hydrologist at heart, and so I guess we're inching towards a fair distribution of water. Pillars of hydrology are floods, droughts, and a fair distribution of water. And this, I think, would be an important step in that direction. So let me first start chatting a bit about something on which, in particular, Ignacio Rodriguez and myself have been working for like 35 years at this point. We studied at length the substrate for ecological interactions, the fluvial landscape, because one of the issues that we keep having in mind and echoing one of the organizers of these winter schools, Simon Levine, the importance of whose work can never be overestimated in ecology and beyond, is that although natural ecosystems are characterized by a striking diversity of form and function, quite a few times, these often, they exhibit deep similarity, deep structural similarities. At times they emerge across scales of space, time, and ecological complexity. So we wonder whether certain universal features that appear in the dendritic structures, like the ones that I'll be pitching in for the fact that are statistically the same regardless of climate, vegetation, exposed lithology, you name it. There is a self-organizing process beyond this, which is super strong, and self-organizing in the sense that it will generate realizations that are statistically identical, regardless of, there's no celestial tuning of parameters that allow them to happen. It happens nonetheless. So it's not a critical phenomenon, but it's self-organized critical phenomenon. Now this, for those of you that have been at ICTP for a long time, know how important it has been in the cultural activities there. So let me talk to you about the river landscape and why, in fact, this is going to be brief but heartfelt. We have quite a few tools. This is taken from, again, Google Maps, and what you take a picture. What you can do nowadays, you can filter and realize what vegetation is, and you can actually expose the terrain to the point that you can have LiDAR or LEADER, whatever you want to call it, maps, in which essentially you have the size of a stamp, with these centimeters at this point. Once you have filtered the vegetation, you have the surface of the terrain in an exquisite detail, which you see in this case is exactly the same bend of the Amazon. And the accuracy on the vertical direction is now becoming more than enough to generate description of details in geomorphology. I've really changed the ball game forever. And this, so this is another picture at the same time. Once you filter the vegetation, the detail of geomorphology that you're having there, objectively manipulated and automatically retrieved and remotely retrieved, it's really phenomenal over a range of scale which is unheard of. So essentially what you have, I'm still attached to the old picture, the first one we took out in 1989 or 90, whatever it was, etc. This is a small catchment in which the restitution was of a few 100 square kilometers, and this was given the pixel size which you have by general tools. It will be like of the order of this kind of 30 by 30 meters, and the accuracy in the third dimension was of the order like half a meter. So what happens is that essentially you have a surface remotely acquired and objectively manipulated that tells you how you can calculate gradients. And gradients means steepest descent direction. So you have a field z sub I, which is a scalar, right? You can calculate a vector, which is the gradient, that is the steepest descent direction, which by all means in hydrogen geomorphology makes sense to assume that the strongest force is gravity by orders of magnitude. So steepest descent direction is also the direction of the flow direction. So aggregation paths can be devised directly via the shape of this kind. I mean, I'm essentially engrossing a particular of that. So you can delineate a line of this kind. And we have a trick, which is a useful trick, in fact, of using a scaling relation for tracing the black line that shows the mainstream in here, which is a well-known Leopoldian rule for the scaling of the width of a river channel. This is a trick for showing how the system behaves, but certainly it has a major implication. You can calculate elevations, you can calculate Laplacians, like let's say essentially the average over the nearest neighbors in a place, so you can easily decide what's the sign of the system, that is, whether the surface is concave down or concave up. You have some sort of tricks to the point that we assume early on, and essentially because of a statistical well-worn argument, essentially our theorem is that square root of 2 is equal to 1. That is, take as a gradient the steepest descent direction, although exceptions can be generated. So essentially you extract the river network from data over the scales around the order of a meter, even less than that now, to the order of thousands of kilometers. Still, we are talking about the so-called runoff generating part of a landscape, where in fact you do have aggregation, you don't have a transportation zoning, we have no significant injection, but still you're talking about 6 clean orders of magnitude that can be ground truthed quite easily. And quite a few at the time we used to go at the physics conference, in particular in Trieste. And it seems that the data set over which we started learning how nature works, in fact, hardly find a match than in the case of a fluvial basin. So it's something we know fairly well. So essentially, we define the master variable for this, which is total contributing area at the site. Like in this case, you have 12 connected pixels upstairs. That is, you essentially have, and it's a typical equation that you had in the aggregation process with injection. If you assume that 1 is the size of a pixel, whatever square meters you have, I have a connectivity matrix in the system, and I'm getting to the point that now a system of design, this is a, well, I skipped an important issue, which I shall return. I'm assuming that this is a tree, that is, in every node, you have a unique path leading to that node by different directions. You don't have loops, essentially, and I'll get back to that. Well, it's also historically quite important because that's the assumption that was made in the first manipulation of a system as we see it, and yet it proved a lucky shot because we didn't search for like optimal configuration of some kind by searching loopy structures, and it turned out to be a fluke. But anyways, what you have is that a WJI means whether it's something which you have a 1 if this J is connected to I, I'm sorry, if I is connected to J, and 0 vice versa. That is, in this case here, you bring in a guy who carries a weight 1, a guy who carries a weight 1, and a guy who carries a weight 1 plus 1, that makes 4. If you move here, you add 1 plus 4 plus 3 plus 3 plus 1, and you get 12 pixels, etc. And this means that this is a quantity which introduces, and the statistical physicist among you has spotted immediately, this is a non-local interaction, which is applying locally. And this has plenty of consequences, of course. Now, what is interesting is that this is a tree. This connectivity matrix has all 0 eigenvalues, so you know that immediately. So if by any chance you try to perturb this configuration generating loops, you would know immediately with elementary numerical checks. A source has area equal to 1 and the like. So 1 is the basic scale, which is delta squared, the size of this guy here. I'm sorry, I trivialized it a little bit, but it shows how we can, in fact, show a remarkable capability we have to remotely acquire and exactly manipulate description which are super accurate of natural landforms over, I would say, up to 6 orders of magnitude. And what is interesting here, I removed the scale bar. And one of the main tenets, and that's the book where Ignacio and I wrote years back, which was were received, in fact, is still used in quite a few even graduate classes, or for its well cited. So what I'm saying is that if you remove a scale bar, you really don't know whether this is a large or this is a small catchment. This could be the Amazon, it could be a small creek in the Dolomites nearby, because nature tends to produce those shapes in a remarkably similar shape, regardless again of climate, regardless of vegetation cover, exposure lithology, you name it, or any kind of perturbation that the system has. How do I know that? Well, actually, we can specialize a little bit, a little bit more geomorphological extraction, the geomorphological consideration of how to extract the proper channeled part of a landscape. For instance, this is the network that connects all the concave sides, the one for which nabla square of zi is larger than 0, concave up. So eliminate the dots here that are the concave sides, which has a well-defined geomorphological reason for happening. The concave sides are called colluvium in geology and geomorphology. Or you can take a subset of that, which is further refined by some sort of a criterion with a threshold that you had in there. You can read in that signatures, for instance, of past climates, I'll see in a minute. So essentially, if I enlarge the thing, you have the channeled pixels, which are the proper parts, which are part of the domain of a fluvial domain, river networks as ecological corridors, as we say. So again, this is what we have seen. And the key result is that you take this as a master variable, that is, you consider that the total contributing area at any pixel, at any site, is a random variable whose probability distribution, in this case, by no chance, we use a probability of exceedance to avoid problems of binning, as you may know, or which we may return in case, is essentially proportional to this value a to a power, so infinitely popular terms like power laws, which is essentially dictating what Simon Levy was saying about the deep structural similarities that emerge across scales of space, and in this case, not in time, but equally well if you have activity on the network. So essentially, oops, sorry, going in the wrong direction. So this is the what happens. Of course, if you take sub-catchments, you sub-sample within a basin, you will have that the maximum area will be limited to the maximum size of the sub-basin you have. But what you see that if you take nested sub-basins, bigger and bigger, you see that the argument you had in the system, in reality, you base an argument which is called the finite size scaling, which is something which is well known to statistical physicists, in which you see that essentially you can have these curves collapsing directly onto one another, showing that, in fact, the deep and remarkable similarities emerging across scales is a well-established fact over which there is a significant literature. Consequences that the system, so it's a well-known fractal, if you have signatures, etc. The geometrical language, as Benoit Mandelbrot, whom we miss a lot and to whom we pay respects all the time for these visionary ideas, it's a common name for that. So it's the language that nature speaks. And what you have is that if you take, for instance, gross features of a domain for interactions like transverse and horizontal length, you have a relationship like the probability distributions are with a remarkably consistent coefficient of less than 0.5, highly non-trivial, or the lengths to the source of any point are well codified and well known. And linked scaling exponents fully characterize the network forms, in fact. So these are, there's a zoo of cases that have been investigated in the early 90s, and this is a remarkably robust result that we've shown. Now, the finite size argument also allows you to get interesting features. For instance, if a finite size argument applies, the ratio of consecutive moments of a finite size scaling distribution produces a particular relationship which shows that regardless of the value of the subsequent moments that you're using, you should have different, the same exponent essentially. So this is a particular kind of a fractal machine which allows us to calculate the elongation of a catchment, which is something which without math, but that Mandelbrot spotted on earlier. And these are the data. It's for you to show what happens, etc. So essentially, a consistency across scale, it can be done because you can use these to generate, for instance, variations in time of the features that generate the network. But what we really know at this point is that essentially the spatial imprinting then generates total contributing area, which rightly called the master variable for the system, is what generates something like, this is what happened to Mount St. Helens after a few minutes after the first rainfall, in fact. So the imprinting has been like canyon-like, but it would stay forever, the planar imprinting of the system. That settled the issue. Or if you take, for instance, Martian landforms, if you apply to the same things, etc., what you seem to be, or what you see, in fact, as something fluid has to be operating in eroding those surfaces. But on this, I shall not insist, even for saying that we have tools for, and then I'm back to the, not really fluvial, but tidal networks. You can tools in which, by studying the landscape, you can extract landforms of a very fine scale indeed. And so these are many of the things that we've been studying for years. Or for instance, if you take like, this looks like a photograph, but it's a digital terrain map of an accurate one, in which you can actually start devising, or whether you can discuss whether these are natural or artificial forms, in fact. So that's what we have. Again, this is a digital terrain map for our treasure, our data set, that you can in fact show how nature works from, again, 10 centimeters easily in the fluvial landscape to thousands of kilometers. And that opens the thing to, I still allow myself to have like 5 minutes on networks, and then I move on to the first exploitation of that. Would be whether all trees are equal. And the idea is that whether the loopless property is particularly distinctive is something also important. And we say why comparing networks is important. For instance, I'm claiming that these networks, not this one, but the three of them, for instance, have the same topological properties. And what distinguishes them are metric properties of a different thing. But it doesn't take, I mean, a scientist to see that the Peano network, which I'm showing here, is different from this one. But they are topologically identical, indistinguishable. So this deserves some extra thinking. This is Peano network, which is something on which we worked a lot, and it was essentially devised by Mandelbrot. Is an exact fractal. You have a hell of a lot of properties which are hydrologically and geomorphologically relevant that can be solved exactly for this construct. Essentially because they map multifractality, for instance, exactly. It's a binomial multiplicative process. In this case, it's characterizing something which is very important for hydrology. It's a benchmark. But again, the topology is the same with the networks, so it allows you to get exact solution for properties that are topologically dominated. And we'll see next class that there are a few. When I'm spending some time on optimal channel networks, which is something which we invented in National, and this is a tool through which you essentially calculate a spanning tree. A tree has to be, in fact, over a given domain. In this case, square, but it could be anything. It could be with boundary conditions, which are periodic boundary conditions, whatever you want to have it. And so the idea is the following. There is an exact statement taken from the general landscape evolution equation, which is nothing but the mass balance of an elevation field Z, in which you have that in steady state condition and the small gradient approximation. And in the case of reparametrization invariance, in fact, you get that there exists a Hamiltonian of the configuration of the system which you're seeing here, which minimizes energy dissipation, which is essentially related to the master variable, the non-local variable at any place raised to a power gamma. Now, on this, I shall not spend time because this is well digested. If you want, you can find the exact solution in a certain places, etc. But what I'm saying is that what is relevant for us is that there is a zoo of possible cases that we know of. For instance, if gamma is equal to 2, these are called random resistor networks that have been studied by engineers in particular for a long, long time. S is the set of, the configuration of the system is essentially the number of L square sides, the total contributing area, which is embedding the connectivity structure and the aggregation level. Now, in the case of gamma equal to 1, this is a very interesting thing. These are called the well, mapping the so-called abelian sandpiles in self-organization. That is, you essentially have a network which minimizes the mean length of the outlet. And what is interesting is that all directed networks, in fact, have the same energy dissipation if gamma is equal to 1. But mostly interesting is that if you assume that this is a problem in which you multiply flow time a gradient, like you have normally the power dissipation, the energy dissipation, as you want to call it, then A is proportional to the landscape forming discharge. It's a well-known result in hydrology. And the slope is proportional to area, to a power which is less than 1. So essentially, you have a class called of optimal channel networks that obey this property. Instead, if you put gamma equal to 0, you have spanning networks, and it's a different ball game in its own. All I'm saying is if you keep going in the system, what you have, in fact, in some case, an issue is that from any initial condition, you can essentially rearrange the system by disconnecting a place, getting a new tree, going to other direction, and checking whether they, well, you can actually find whether you may accept changes in this case, which is the basis for this procedure. Or you can put some probability a la Gibbs, like simulated annealing, and you can actually generate these figures here. Now, an interesting point, and I want follow up on that, is you can calculate in the case of trees the true thermodynamic entropy. That is, you can count the log of the number of states that have the same energy, which scale with a certain condition. And total energy dissipation scales with a different exponent, which is larger than 1. So in the thermodynamic limit, if the size of a network is big enough, energy dissipation is also minimizing thermodynamic entropy. So these are the figures that we can generate. And in fact, you can show that if you start making good comparisons, you can calculate fairly well how these exponents are perfectly matched, in fact. So matched scaling exponents are the true tool for comparing different trees. That's it. Also, an interesting point that by another lecturer that you have, Amos Maritain was the lead author and a former ICTP professor of a ground state, the scaling property of a ground state for this case. And what is interesting, in fact, the scaling exponents for the ground states are not the ones we observe in nature, nor the ones that we get in the case of so-called feasible optimality. That is optimality, which is optimality, which is dynamically accessible. Randomness is not a thing. So essentially, this is an Eden growth, which is an entirely random dominated choice. And if you make a good comparison of these two networks, look at the different boundary conditions. They look similar, but if you look carefully, they are not, like your eye is telling you. And these are the two different conditions. These two scale differently. They are both network, and the chance that the system has to reach the ground state is nil in practice. And this can be seen progressively by relaxing boundary conditions, size, and the likes. So these are the typical networks over which I shall be talking about. And I will jump now to the, I'm skipping details essentially on the fact on why, in fact, networks are, I may get back to that and later on in one of the following classes. I think it's more important that I move on, but the idea is that I hope I conveyed, that can give you the material on that, is there some coherent thinking on how, in fact, we have similarities, structural similarities emerging in the substrate for ecological interaction, emerging across scales of space. So the first thing that we had in mind is using on a substrate of this kind, the neutral theory of biodiversity. Why neutral theory of biodiversity? You may know that the neutral theory was originally proposed in complete analogy with the neutral theory of molecular evolution, which assumes that gene mutations are selectively neutral. That is, new genes are demographically equivalent to the old genes. And they do not give any advantage in terms of decreased mortality and or increased fertility. And it's a paradigm that dominated the molecular processes and still does for a long time. The main advocate, in fact, for a neutral theory of biodiversity was a colleague from Princeton, now at UCLA, Hubble, who wrote the fundamental book of the neutral theory of biodiversity. That is, he was asking at whether the idea, starting from works done on tropical forests, in which essentially he proposed to have mutations replaced by the occurrence of new species in a landscape. So the idea is that it was a revolutionary one, because essentially what you do, you assume that all species are considered equivalent at per capita level, and on which I shall build more in lecture 2. But what I'm showing you, and hopefully I will convey my main idea, is that if you have a neutral process over a particular topology of a substrate, this has consequences. So the conditions for a species to occupy a site, for instance, and maintain a population are the dispersal ability of a species, the habitat suitability, and the susceptibility to any kind of biotic killing of any kind, etc. But we want to see, okay, now, of course, we have conditions to grow and maintain a viable population in a place, but what happens, that was the key questions, knowing that we know a lot now about the recurrent characters of fluvial landscapes, in fact, which have a spanning tree over which interactions occur. What would be the consequences of the extension of the neutral theory of biodiversity to space-explicit ecological setting, but in the particular case of a river network? So essentially, the experiment I'll be showing you now to introduce the following discussions is one in which you have essentially, or suppose you have a lattice, okay, which in a hydrologic term called savannas, because essentially what you have is that every single node is allowed to interact with the nearest neighbors. Whereas in any chunk of a river network, you can have two sites that are neighbors, nearest neighbors, but they are completely, connectivity-wise, they have a very long distance from one another. So essentially introduced, because of a directional dispersal, which is embedded in the network structure, with or without drift, because that's the point, you have a system in which the connectivity matrix is a completely different picture. So we can apply the spatial ecology sensu, like Tillman, the famous work with Kareva in 1997, is extended in the model which I'll be discussing now, extended to embed the topology of the substrate. So this is what happens in setting the simplest possible model. So you have, assume a distribution of colors, and assume the color is a species. This is what in social sciences, statistical physics was called the Botar model in the beginning. So assume that at random, you kill any existing color at any place, and you have two options. With probability ni, you replace it by one of the colors that is not existing in the particular place. And with probability 1 minus ni, and this is a very small number, you assume that in this case, the color that will be taken out of the piece will be the one in which all nearest neighbor, the most abundant of the colors in your nearest neighborhood. Now, this is interesting because you assume that there's no stronger species than another, right? So you assume that if this is a political opinion, there's no political opinion will be stronger than the others, and will be simply what you actually have as it's the wisdom of a numbers, of a large number of you have in the system. So in these, every now and then, like every 10 to the 5 trials, etc., the point you choose at random and you kill is just parachuted from outside the domains, mimicking either speciation or migration or immigration from different places. So the only difference between the two is essentially the topology of a connection. And when we saw this, we got kind of excited because we said, look, what happens is that the only ingredient you have changed is the matrix, is the neighborhood, the definition of neighborhood. In one case, the one you see on the left was open and thereby more open to interactions. And the second one was more protected in a sense. It was due to the presence of the network itself. Now, what is interesting is that if you just make it quantitative, which you have it here, you essentially have, not only you have the different degrees of irregularity in the boundaries, which is proposed, but in this case, which is the network system here, the neutral system here, what is clear is that any statistical measure that you have of the distribution of the colors is essentially completely changed. And the rank abundance curve shows that the biodiversity of a network system is much bigger. And of course, the first thing you say, okay, is this a realistic model, the neutral model of a fluvial ecosystem, of a fluvial stream microbial ecosystem distribution or not? Well, it so happens that patterns may not be, neutral patterns do not require neutral process, perhaps, or equifinality issues can pitch in. But the fact is, this was an intriguing suggestion early on. It happened in 2009, before we embarked in this study, etc. But for us, it was worth checking. Why this is interesting? Because this is a result that it's absolutely robust with respect to a number of generalizations. Namely, this is 1.1 species. But this could be evolving into a metapopulation model. It's one node because a local community with a certain number of species, the same rules of engagement for the interactions. And you can also have is that you can relax the possibility that it's only the nearest neighbors giving you by sheer majority the new colonizer in a place in which you don't parachute the new species. And the result is the same. So a dendritic substrate for interactions changes completely the ball game, regardless of a number of assumptions. Still, in the obvious assumption that you're assuming that all colors are equally valid, and you're assuming that all species are equivalent to the per capita level. But we thought it was interesting. And the first thing that we did, we applied the same rule, individual-based, or meta-community-based, but with a kernel, that is the radius of influence of the area over which you average progressively to get the majority replacing the place. Could be immediate neighborhood or mean field, that is the one you essentially go and check what happens in the system. You can do that on a 2D lattice. You can do it on a network. You can do it on a 3D lattice, on a 3D network. And much to our surprise, what have we found? Okay, one good measure to see what happens in this case would be essentially related to how long would a certain color last from onset when it's created or in a particular place, or it has local extinction. And this is called the persistence time of a color or of a species into a landscape, only in this case affected by technicalities like the structure of a network. And much to our surprise, the distribution, whatever you want to call it, certainly there is a huge range of scales that covered by power law in this case, show that there is a distinct and unmistakable effect of the topology of a substrate for topological interaction. So in our case, we took on to a first empirical check, because it's an interesting thing we did too, in fact. So we studied we had like 41 daily values, 41 years of observation of breeding birds in North America into a place. And here you see what is this, the persistence time in this case, meaning suppose that you are in this area here. I should have, yeah, here it is. I should have in this area here, you count in those observational sites, you count how many times here you spotted for the first time a particular bird, and here for a number of years you haven't. So these are independent persistence times. And you can study the value that you have at different level of aggregation, for instance. Or you can do the cancer spray herbaceous plants, etc. So you can characterize the thing. It's long story short, but what I'm saying is that you can isolate the effect of a finiteness of a sample, which you have an atomal probability if you have a species that's been continuously observed, for instance, and you can characterize this interesting feature. So long story short, what you have is that you confirm what we have seen. Topology means a lot, and topology of a substrate for interactions has an effect. And to the point that you start, for instance, aggregating in different spaces, you can transform this scaling effect, exponent of a lifetime, into a distribution of biodiversity. So essentially, by taking coarse-grained versions of the same space, you can find the probability distribution of the survival, how it behaves under coarse graining, and thereby generating a true species area relationship. And the next step over which I want to show you, and on this I'll be relatively fast, it's only the last 5 minutes of a class, we said, okay, say, look, of course, and you talk to an ecologist, say, hey, but this is bullshit. I mean, species are not equal at per capita level, how you can do that, etc. And yet, we thought early on there was something manifest to be completely false, a topological effect of a substrate on the nature, the recursive nature of a form and function, which is embedded in the structure of even networks, regardless of whatever. It is truly a universal feature. So we decided to test on, in my lab, these with living communities, in which there's nothing neutral. There is absolutely nothing neutral. So essentially what we do is just, we choose, for instance, you see how rudimentary the scene is. We should have had the money to buy a robot, and in the absence of robot, you have, so this is essentially what we have in the lattice, in which nearest neighbors are the 4 nearest neighbors. I mean, in which nearest neighbors are the topological ones, the adjacent things, and the structure in which these 2 guys are not nearest neighbors. And we did that by brute force. This is Francesco Carrara, now at ETH, and this is Enrico Bertuzzo, now a professor at the University of Venice. Now, long story short, this is what we got. Replicas were as many as needed, and this is the experiment that you have in the random network system that you have, or in the thing that you have. And the color, essentially, is a measure of species richness. So in this, the theoretical idea was telling us, look, guys, there's something which pertains to the fact that you have a certain topology of the connections. It's regardless of everything else. But the prediction came directly from a neutral model. If you have like a lattice of interactions, this doesn't happen experimentally. And this, again, this is a living communities. I'm talking about, in this case, I'm giving you the details. I took a note someplace here about what is the experimental community. I'm talking about 21 protozoan species, in which we had, 9 protozoan species, 1 rotifer, which is a multicellular one, and a set of freshwater bacteria as food resource in the place. And the dispersal was done manually, but it was done essentially according to the things, etc. And I can give you details if you care for. An interesting tool was also published in another set of painfully conducted experiments, in which you essentially get, okay, now suppose that we take into account the fact that habitat capacity in a river is hierarchical, okay? So essentially you assume that the medium in any single well depends on the number of upstream sides. So you have a hierarchical structure. Or you get system in which the same total amount of medium is randomly distributed, or in which you take a uniform distribution of the system. Believe it or not, the hierarchical system is a completely different structure. So, and that's the take for the next classes, saying that whether neutral models or models of anything else, modeling of riparian vegetation, modeling of species diversity, etc., we're talking about the landscape quite a bit. When you have an environmental matrix, which is made by nodes and connections, the substrate matters. And I'll show you how this, in fact, has a say on the oriented graph is something which we give. We don't assign a network of interaction. This is given by the geometry of the system. And in good timing, in fact, I'm showing you the conclusion, a general conclusion, but essentially what I have shown is that eco-hydrological footprints of rivers as ecological corridors were suspected from early abstract theoretical work, and confirmed by empirical analysis on a particular set of persistence times measured for breeding birds or plants in the Kansas prairie, or experimental work done on living communities in this place. So I'm ready for your questions. Of course, in the following classes, what I'll be doing, I'm talking about species, about the biological invasions. I'll be showing you why biological invasion is slowed by bifurcating structure of the system. And then I'm talking about disease, which is an interesting point. Disease is tackled in this manner, in a spatially explicit manner. Okay, let me check. Well, I had chats. Okay. Hi, everyone. I'm having serious problem with electricity supplies. Oh, no, I'm sorry. That's a question about the electricity supply. I cannot help you. I'm sorry.
So we are ready to take questions from the audience. If you want to speak, just raise your hand in the list of participants, and I can give you the floor. Okay. We have a question from Miguel Rodriguez. Miguel?
Hello.
Ciao, Miguel.
That was a fantastic lecture. Thank you. For this last part, where you do these experiments with microorganisms moving from well to well, will that pattern be robust to different degrees of base diversity? Will that be true for much larger values of richness, for example, in those?
Okay, that's a super question. Super question. Now, well, we have experimented with a varying number of protists, in fact, because we needed to have a sizable thing. And I have to say, the protists have been a good model organism, in fact, for these kind of studies for a long, long time. And if you look at what there's a number, Hollyoak, Florian Altermatt, which was actually working with us on these, etc. They work a lot on so-called dendritic substrates for interaction. But what the hell? The dendrites were just a pipe and a few bifurcations. That's not the river network. That's why I keep saying, and I think I convinced Florian, who followed us, etc., to the point that in fact he put on, you'll find a note, he put an open source code for having his own networks in the place, because you have to have the number of sites that you have are constrained by the total area. It's not that you can assign them independently. The connectivity, which is suited to a fluvial ecosystem, is a specific one, highly constrained one, a highly recurrent one. It's not one you just had like a pipe and a few branching patterns in there. And so the question is that we have experimented with different number of processes, but we haven't explored it a hell of a lot. All I'm saying is that, as you will see, soon after these kind of confirmation, we got more and more enthusiastic. I got an ERC grant, and we started having lots of people working on different aspects of that, about which I'll be talking in the next two classes. And I'll be showing how we did species, we did population migration, which is a fascinating subject I'll be talking to you, because we start from migrations, human migrations in the 19th century. It's a long shot. And then I'll be talking about disease. Believe it or not, with the same tools, we started simulating spatially explicit models of cholera. Well, waterborne, of course, it has to be, right? The pathogen, and whatever has the pathogen living in it, or proliferative kidney disease in fish. So anyways, we haven't experimented a lot more in the lab. Mind you, at the time I didn't have the money for the robot. So Francesco and Enrico stayed up long nights in pipetting from one side to the other. And that's typical, you know, you see, typical professors say, what are these details? It's not detail, it works a lot.
Okay, thanks. We have a question from Susie Barughese. Do you want to speak for yourself, or do you wish us to read your question on the chat?
Susie, I see it. I see it here. You want to repeat it, Susie, yourself? Otherwise, I read it.
You can go ahead if you can understand the question.
Okay, the question is, are there evidence for biodiversity reduction whenever rivers are diverted or dams or construction are constructed? Of course, fantastic question, Susie. Yes, there is. In fact, a group of fish biologists in Spain, it's Pepe Barquin, I think, is a guy in Santander that published quite extensively on the study of how biodiversity is impacted by, it's actually common sense. It's common sense on a number of counts. For instance, I could tell you that there are a number of studies, especially the biofilm guys, are very prominent on that. If you modulate the natural sequence of stream flows, you change the ecosystem, period. That's a fact.
The corollary is like, can we use it for example, impact studies prior to constructing dams to say that, you know?
Oh yeah, but I haven't done it, but the Pepe Barquin and his group has done it quite a bit, actually.
Thank
But,
you so much.
I mean, it just, but even in common sense, I mean, it's known by what a dam does, besides the interruption and the change in connectivity, which is directly reproducible in the case of this, because essentially you change at any downstream point the total contributing area, right? But the sequence of stream flows is not proportional to area anymore. So essentially you change the structure of the engine for interactions. And I mean, it's known, people that study the biofilms know that, which is the basis, the engine for the ecological interactions in the microbial ecosystems or fluvial microbial ecosystems. And so there is evidence for that. Yes, the answer is yes. I haven't done it. No, I haven't done it.
We have a question from Vitor Sudbrak.
Okay, within this model, the river capes, could we add the effects of structural changes in river curvature due to coastal erosion or river, okay, or rivers driven by deforestation or riverside vegetation? Okay, there are two different issues. First, okay, it's two separate issues. To the first one, the answer is no. Why? Because the recurrent properties we see in rivers do not apply generally. They apply the so-called runoff producing areas. So the river structure is essentially the runoff producing area where the landscape form and discharge is proportional to the total contributing area at the point, the non-local quantity. Then, for instance, if you enter the desert like the Colorado River does after Glen Canyon Dam, for instance, then you have a place in which you have no injection of water. So essentially the transportation thing is dominated by geomorphological process and morphodynamics, which is not related to the aggregation process at all. And then you have the delta of the estuary, which is the distributary system, which is again governed by different rules. What I'm looking here is that the runoff producing areas, remind you, is not the whole of the river basin, but it is something which can easily extend to scales of the order of thousands of kilometers inside. If you're talking about deforestation of riverside vegetation, of course, yes. And I work on that. I'll be talking about briefly about models of vegetation, over which we worked a lot, actually. So the answer is yes to the second one. Is that okay, Vitor? Oops, do you hear me?
And we can safely assume that Vitor is satisfied. Yes,
Oh, yeah,
he is.
yeah.
Okay,
Oh, yes.
do we have any further question from the audience?
Can I ask one more question?
Yes, please, go ahead.
In your persistence time distributions patterns, you show that these networks, these river networks, sit somewhere between 1D and 2D fully connected grids. Will these be predicted by the dimensions in that fractal structure?
Absolutely. That's again a very good comment. What happens is that it's the topology of a substrate that matters in determining the persistence time. So you sit in a point, you close your ears, and you measure for how long the color stays there. Okay. You find that if you do that, of course, every site is a different persistence time from the onset of a species. We're not measuring abundance here, so we don't measure the number of colors. So color red in this place, yes, for how long it stays, and you measure it. So every particular from onset to local extinction is one value of a random variable persistence time, local persistence time. You do that for every single thing. But what I'm saying is that the game of simulating that, in a couple of hours you can do that, because it's absolutely trivial, right? If you give by hand, for instance, the connectivity, which is slightly more complicated, but okay, but the essence is that one. What happens is that what you see is that depending on the topology of the system that you have, you don't know anything of what's going on in the model, etc. You simply count. And you see that those distributions, once you start making big enough to have enough statistics, etc., behave completely different. And that is the counterpart in which this is telling you, look, there may be something deep going on here. Because what we are saying is that depending on how I'm connected, it's how I'm exposed to interactions. And how, in fact, the distribution of my attention is completely different, changes radically. And what is super interesting is that this topological effect has been, it's singled out exactly, in fact. But what is interesting, if you look at data, it's a tricky thing, because If you look at the data, for instance, look at breeding birds in an area like of Georgia, God knows what. You know, Audubon Society started like 50 years ago in collecting those. The network of the bird watchers is a serious business in the North United States, and meritoriously one. What happens is that what you had is that, okay, let's suppose the cardinal that was seen in this area now, that it hasn't been seen like this year, he has been spotted. Next year, he hasn't. So essentially, you measure what happens locally. And then what you do, you can make it a bigger area. So it changes because it may be local extinction, can be here and not in the nearest. So at the continental scale, okay, if you assume there's no migration of cardinals, etc., it's speciation that you're measuring. It's you can call it a speciation rate or a true speciation rate. So what I'm saying is that you can empirically, there is also an issue because if you have a finite sample in terms of length, what happens is that you may ask yourself, okay, look, if you see throughout always this, you always spot the same bird in the same place, you have an atom probability of 1, okay? And that is very important because you see how this atom is reducing with the size of a sample. It's telling you a hell of a lot of the underlying distribution. And so being careful, but you see, I'm showing you in 45 minutes, like 4 or 5 years of work. And but again, if you care for that, I'm ask your library to buy the book because we spent a hell of a lot of time in putting it together.
Thank you.
I think we have time for a couple of questions more, and then we'll close.
Okay, Tuan is asking, could you please remind me what is the difference in topological structure between the savannas and the OCN? Okay, savannas means that in every site, your nearest neighbors are the 4 nearest neighbors in the coordinate direction. That is north, south, east, and west. That's all you check for, okay? So once you kill in a place, a system, you replace the color you have in there by the most abundant color in the nearest neighbors. And if all 4 values are different, you choose at random, okay? In the case of the OCN, you have a directional dispersal. So you have essentially drainage direction that will tell you who's connected with whom. So you may easily have that because of this directed structure, nearest neighbors topographically may not be nearest neighbors in terms of interaction. And that's the drainage direction, which you get directly from the landscape elevation. There's another one. Zebsa, Rabat. Always I think that there is a limit number of species that coexist in structure. And then all about spatial structure. That's a good question too, Zebsa. What I'm saying is that I'm talking about the abstract model now, in which one site is one species, okay? But you can generalize this into a meta-community model, which I'll be explaining next class, in which for every node, you have a meta-community, a local community. It is a community of communities. So you can actually calculate carrying capacity. If you're worried about that, you can calculate logistic curves, etc. You can put this into a spatial ecology framework, like the spatial ecology framework that Tillman, the mighty Tillman and Careva put together. Only thing, what we provide, it's those recurrent features of channel networks, which you know better than anybody, because we worked for 20 years on those. So the answer to your question is, of course, there's a carrying capacity on each node. You bypass it, but you see the pattern explained by the individual-based, say kernel 1, that is only nearest neighbors thing, etc. It's telling you a story which is confirmed by making the kernel, say, mean field, like the entire structure of the catchment, or number of species fixed, or number of species variable, or whatever. The result, that is, the pattern is affected, resists, and is general. Thank you.
Okay, so maybe it's time for us to call an end to this first lecture. And thank you, Andrea.
Thank you so much, Antonio.
For the beautiful lecture. As usual, we have the opportunity to participate into the breakout rooms where you will be sent automatically. And otherwise, we meet again at 3:45 for the next lecture of the school. Okay. Thank you.
Ciao, ciao Antonio. Ciao, ciao.
Ciao. Quindi ai nostri diploma facciamo fare il tuo, quello di domenica e quello di San Benedetto. Perché non fa una cosa in più rispetto a quello che fa. Ah, ok, ok.
Ué, a Isabel segue uma página de Umbanda.
Okay, so welcome back. I think that everybody has joined again the main room, and we are ready to start with our last lecture for today by Stefano Lesina from University of Chicago. Please, Stefano.
Good morning, good afternoon, and good evening, everybody. So this is the second lecture on the theory of ecological assembly. And just to recap what we've been doing, like the first lecture, we've been exploring a little bit of the history of the problem of assembly in ecology. And then we looked at some features of the model that we're going to use for our exploration, which is the generalized Lotka-Volterra model, which is arguably the simplest model for population dynamics. And just to give you a very brief recap of what we looked at. So this is the form of the model here. And it has just like the parameters are just a vector of growth or death rates and a matrix of interaction A. And then we looked at the notion of an equilibrium, which is like a point, a certain density at which like dynamics are fixed. And we saw that like this equilibrium, if it exists, it's unique, provided that A is not singular. That we call this equilibrium feasible if it is attainable by an ecological system, meaning all the species have to have a positive density. We saw that this equilibrium is the time average of the dynamics in the long run. And then we saw also that the feasible equilibrium is necessary for coexistence, is not sufficient. To get toward like sufficient conditions, we looked at stability. And in particular, we looked at local stability, meaning is this equilibrium gonna be, you know, like would trajectories go back to the equilibrium if we slightly perturb them away from the equilibrium? That's like a local stability. And we saw that this is very simple to do because all you have to do is to form this matrix, which is like the diagonal matrix with the equilibrium on the diagonal times the matrix of interaction. If this matrix M, that is called in ecology the community matrix, has only negative eigenvalues, or better, eigenvalues with negative real part, then the equilibrium is locally asymptotically stable. We also introduced a much stronger form of stability. It's called global stability, which simply says that if I start the system with species with all positive densities, eventually the trajectories will converge to the equilibrium. And to prove that, what you have to do is to find a matrix C that is diagonal and positive, meaning only positive coefficients on the diagonal, such that this new matrix, that is a symmetric matrix, that is C times A plus the transpose of A times C, is stable, is negative definite, right? So if you can find that, then the equilibrium is globally stable. All right, but we haven't really got to the point of talking about assembly. So that's what I want to do today. And in particular, what I would start with is like the reasons for which building a general theory of assembly is so difficult. And for this, I'm going to follow quite closely like an introduction that we have in this paper with my former student, Carlos Servan, that possibly will be published next year. So we argue that there are three main issues with assembly in its general form, right? So imagine that this assembly is just a process by which some species enter a system, they interact with the local community, and then something happens. For example, the species established, send some other species extinct, or whatnot. The first problem that I would like to highlight is that of invasion rates, right? So if you think in this system, we basically have two different timescales. One timescale is the timescale at which new species join the system. That's like the invasion rate. And then we have the rate at which like dynamics evolve, right? So you can imagine like, provided that when I put in this species, something will go extinct. How long does it take before they go extinct? And the interplay between these two timescales, it's actually complicating the problem quite a bit. And I think that the simplest case to study is one of like a rock, paper, scissor community, right? So imagine that we have three species of, say, plants, right? Such that each species, like the species, say, rock, can happily stay in its habitat by itself. And I just like use this notation for that, right? And similarly, the scissors plant or the paper plant, they can happily colonize this habitat. What is the problem? That when I put two species together, for example, rock and paper, what happens is that one of the two species will go extinct, right? Paper, win over rock. And so like if I wait for long enough, I start with rock, paper, I end up with paper alone. Similarly, rock, scissor, I end up with scissor alone. No, sorry, this should be rock alone, rock. All right. Interestingly enough, if I start the system with all three species together, they can coexist. They would cycle or do something like that, right? So now if we're thinking of the process of assembly, imagine that we start with like the bare ground, right? There's nothing. And then one species comes in, for example, rock. And then after a while, we go to this unstable configuration of, say, rock and paper, right? Now, if we wait for long enough, we will end up with just paper. But if scissor joins this configuration before rock goes extinct, then we're in this state with rock, paper, scissor, and therefore the system can assemble like to the full community. So you can see that really what matters is the speed at which we have the dynamics versus the speed at which we have invasions, right? Like, so that's one first complication. The second complication has to do with invasion size, right? So how many individuals of the new species come into the system at one point? And here, like the simplest example to think of is like two competitors in Lotka Volterra, where we have bistability, meaning each species is happy by itself, but then when I start with both species together, I could end up with one stable state, only species A or only species B, depending on where I start, right? And so what happens is that if I'm sitting at the state with only species A and I invade with a little bit of B, I go back to just A. But if I invade with a large population of B, then I can kind of switch like to the other half of the plane in which trajectories will lead to B. So the outcome of the invasion depends on the size of the invasion. The third problem that I see has really to do with the dynamics of the local community. So imagine that we've seen like last time, a set of species cycling, right? And so imagine that you're a predator, you have to come into the system and you want to have your prey available to establish. If your prey are cycling, maybe you could establish and invade when prey are high enough, but not when they're low enough, right? So then the timing of invasion would determine the outcome, right? So these three main difficulties, I think, are the three main roadblocks to developing a full theory of assembly. So in our paper and in these lectures today, what I would like to do is to make three strong assumptions that get rid of these three problems. The first, we were saying like the invasion rate. And so what we're going to postulate is that invasion events are rare, so rare, in fact, that the local community, it's always at the state, at its final state, right? Like it's always at its attractor that would reach, like would be reached by the dynamics, right? So what I mean is that if we have the rock, paper, scissors, we will only see rock, paper, or scissors. We will never see the three species together because that would basically require two invasions in a rapid sequence, rapid enough to allow the establishment of the full community before the second species goes extinct, right? This is a very strong requirement. Is in fact even stronger than it looks because like the time of the dynamics could change depending on the composition. This strong requirement and this strong assumption is what it's typically made in all studies of, say, population genetics, or almost all studies of population genetics, right, where we always assume that there's a wild type and only one mutant. There's no two mutants or three mutants at a time. We do the same in ecology when we're performing invasion analysis, right? Like we're saying these communities is resting at its state, like final state, stable state, for example, and now I introduce my invader. Second, to get rid of the problem of size, what we're going to assume is that invaders arrive at very low abundance. So low abundance that they don't feel self-interactions, for example, right? So self-limitation or crowding with your own conspecifics, they don't feel that. And as such, they will only be influenced by like the state of the species that are in the local community. And we will see what I mean by that in a second. And third, to get rid of the problem of the invasion timing, the simplest solution, even though again, it's a very strong solution, is to say there's only equilibria, right? So we can study systems where we know that all the dynamics are given by equilibrium. There's no cycling, there's no chaos. And as such, the invasion timing problem is removed. For example, in generalized Lotka-Volterra, if we have a symmetric matrix of interaction, we will only get a fixed point dynamics. All right. Throughout like these lectures, I will distinguish between two different types of assembly. I call them top-down and bottom-up. And they're quite different in the sense that top-down assembly, what I am thinking of, is a massive invasion of all the species at the same time, which is akin to saying what? I start my system with all positive densities at arbitrary densities, and I see what happens. So in this case, assembly is really like more of a disassembly in a sense, right? Like, so what will happen is that I put all my species in a system. Dynamics will do what they have to do. They will probably send some of the species extinct, so some sort of dynamical pruning, and then I reach my final configuration. At the other extreme, we have bottom-up assembly, where I start from the blank slate, like I start from an empty environment, and then species trickle in one at a time, and then I let the dynamics elapse, and then I add another species, and then let the dynamics elapse again, and add another species, and so on and so forth. Right, so this you could think of like the two extremes of one invasion at a time, bottom-up assembly, all invasions at one time, the top-down assembly. All right. Now the last ingredient that we need to really like start studying assembly is to see what happens when a species invades a very low abundance community that is resting at an equilibrium point. So let's again go back to our generalized Lotka-Volterra, and for example, write the per capita growth rate, right? Meaning like I take like the xi dt and I divide by xi of t, right? So that would be per unit of individuals, right? And so you can see that on the right-hand side, we have Ri, like the intrinsic growth rate of the species. And then we have the self-effect, right? So because this is the first species that arrives in the system. Because it starts at very, very, very low abundance, basically this term here, it's negligible, right? It's basically zero. And as such, what we're left with is just the growth rate, right? So you can imagine that the species will start growing if it has a positive growth rate. And then if it doesn't, it will just immediately go extinct. Okay, so this is what happens when we have the first species coming in. It better have a positive growth rate in that particular environment. All right, and then what happens then if we have more than one species? For example, let's imagine that we have two species. And so in this case, what we have is that there is one species that is resting at its equilibrium point, right? So this would be species I here, that is resting at its equilibrium point. And now we add a second species. And what we do is exactly the same, right? We compute the per capita growth rate that is here, like by taking like dxj dt and divided by xj of t. And now we will have more than one term, right? We will have, as we had before, the growth rate of j, but now we have all the influences of all the species, which will be only like the species that was already there, let's say I, and the new species j. So as before, we assume that the effect of j on itself is basically 0. And so this simplifies the problem a little bit. And because xi is sitting at its equilibrium point, then we have that this whole quantity has to be greater than 0 for the species to start growing in the presence of the other species at equilibrium. Is this clear enough? Right, so this is what we call an invasion criterion. And in fact, we can extend it to multiple species, and we will do that in a second. Just like to make something even clearer, what will happen when this condition is met, right? So imagine that this species 2 can really start growing. Well, it could initially grow and then go extinct, or it could displace the first species, right? Displace species I, and therefore we end up with an environment with only J, or they could coexist together, or in fact, they could even like both go extinct. That is unlikely with the parameters that we're gonna do. All right? Now, we can do the same for multiple species. Of course, this will require a little more algebra, right? Because we have to divide like these interactions into the interactions of the species that are already there and the interactions of the species that are not there. So what is really convenient to do is to divide my vector of the densities, my growth rates, and my metrics into blocks, right? And in particular, you remember that we put x star for a feasible equilibrium point. Now let's put this x bar for an equilibrium point that is composed of 2 pieces, right? The first piece is a bunch of species, say k species, that are resting at an equilibrium point. And the other piece of this equilibrium is a vector of 0 for all the species that are not there, right? So we have split like the populations into 2 classes, those that are already in the local environment and sitting at equilibrium, and then the remaining species that could potentially invade. And we use the same order to write the growth rates. So this would be the growth rates of the species that are already there. These are the growth rates of the species that are not there yet. Then we have to do the same with the metrics, right? So now, because for a vector, we will have 2 blocks, right? but for a matrix, we will have 4 blocks. So A, K, K would be the interactions of the species that are already in the environment with themselves, right? With the other species already in the environment. A, N minus K would be the interactions between the species that are not there and the other species that are not there. And then, of course, the off diagonals are the interactions of the species that are not there with the ones that are there, and vice versa here, right? So now what we need to do to say this species, the system is at some equilibrium, like of the kind that we said, right? Like we first have to have that x of k must be feasible, right? And this is exactly this condition, right? Which only involves the first block on the diagonal here, right? The interactions of the species that are there with the other species that are there. And then we can write a second condition, which we call the non-invasibility condition, which says, in this point, if I try to invade with any of the other species at low density, they just die, right? So that means that this point where we are, where there's k species that are coexisting and the other n minus k that are absent, is somewhat stable, right? Like it's difficult to move away from this equilibrium. In fact, we will see that for Lotka-Volterra, you know, with certain conditions, it's actually impossible. So this will be the end state of the community. And so we can write this condition for invisibility, which looks exactly like the one for 2 species, but now there are several species. And now what we can say is that if we can find an equilibrium x bar such that the k part of the equilibrium is feasible and stable, so stable when considering only the species that are already present, and B, the other species cannot invade this equilibrium, this is called the feasible stable non-invisible solution, or also like the saturated equilibrium, right? And like the book that I referenced, like of Bauer and Sigmund, there's a very long discussion about this. And in fact, for this type of equilibrium, you can show that, again, if our matrix interaction is a little special, such that we can find a matrix C that makes like the symmetric part of A negative definite, then there is always a saturated equilibrium in which like a bunch of components, like a bunch of species are positive, the rest are zero, and this is going to be globally stable, meaning I can start my top-down assembly by starting the system at whatever positive condition for all the species. I always end up with this saturated equilibrium. And to prove this point, you basically use the same type of algebra that we did for the Lyapunov function of the generalized linear model, just generalized Lotka-Volterra model. And yeah, so I'm not going to go through the details of this, because you can read in the notes. All right. So this moves us to lecture 2. So maybe this is a good time to look at the chat if there are any questions. I don't see any questions. All right. So then we start exploring assembly with this top-down assembly, which means we take all the species and we put them in the environment at the same time. And again, we use generalized Lotka-Volterra. We use A that is symmetric and stable, right, with random coefficients, right? So we just build like a random matrix, make it like stable in this strong sense, right, that we have. And then we choose like the growth rates at random again. And then what we want to know is what happens in this case, right? So we want to draw some sort of very generic conclusions on generalized Lotka-Volterra with random coefficients, given that the matrix of interaction is strongly stable and symmetric. And this case was studied by Carlos and some collaborators of mine. You can find the paper here. So let's start with the trivia case, right? I think you know the answer already. So imagine that we have a matrix of interaction that is diagonal. So species only interact with themselves. They don't interact with the other species at all, right? So each species is self-regulating to some extent. So you can fit only so many individuals of any species in the environment. But other than that, species don't care about each other. At this point, what would be our criterion for invisibility of the species? It has to have a positive growth rate in the environment, right? If it has a negative growth rate, it will die. If it has a positive growth rate, it will go to its equilibrium, because it doesn't, the species does not care about the other species at all. If we're saying we sample these growth rates at random from a distribution that is centered at 0, right, with a mean of 0, then we would have 50% probability of getting a species to persist. And therefore, if I just like take a bunch of species, say 100 species, how many species will persist on average? 50, right? In fact, like we know the whole distribution. It's the binomial distribution will give me the number of species that persist, right? The binomial distribution with parameters n, which is like the number of species I introduce in the environment, and 1/2, where this 1/2 is basically the probability of having a positive growth rate. Okay, that was a bit trivial. And so let's start thinking about more complicated models where species do interact with each other. And just to start, to get your sense of what we want to do, let's start with some simulations, right? We take like species that interact with each other. And what we do is like we write a function that draws some sort of random matrix of interactions. You can see here interactions are taken from a normal distribution, makes this matrix symmetric, and makes it stable. How do I make this matrix stable? I just look at the positive eigenvalues, if there is any, take the largest positive eigenvalues, subtract this constant from the diagonal. By doing that, I shift all of the eigenvalues of the matrix to be in the negative plane, right, like half plane. And then we have just like random growth rates that are taken from a normal distribution. Okay, so we start with 7 species. We build our stable symmetric matrix. We draw random growth rates. We start at some random initial conditions, and then we integrate our dynamics with these parameters. And then this is what happens, right? So we started with 7 species, and you can see here that 2 species started immediately going extinct. They never recovered, and then they went extinct. While the other species, you can see that they go to their equilibrium, which is marked here as this dashed line, right? So in the end, we have 5 species coexisting. The other 2 have gone extinct. Now, you can see here that we reach some sort of equilibrium dynamics for this case, and I told you that this is always the case when we choose this type of parameters. And this is what we would call this saturated equilibrium, right? Like some species have positive components here, and then 2 species have a component of 0, right? Like these species are absent and they cannot invade when they're rare. Just to show you that this is in fact globally stable, let's take the 2 species that go extinct and put them at very, very high density and leave the other ones at low density at the beginning. And what happens, it's the same. You see, like they start very high, but they fall down and eventually go extinct. And the other species, they go to the same equilibrium as before, right? So in the end, we obtain exactly the same vector of densities here. So this is one very computationally expensive way to compute, right? Like this equilibrium is to do what? Like we take the system, we started at some initial condition, and then we run the dynamics, we get this saturated equilibrium at the end. Turns out that you can adapt some algorithm that was originally developed in game theory. It's called the Lemke-Hosson algorithm. And you can use this algorithm to just very efficiently find this saturated equilibrium. This works when we have this matrix A that is strongly stable and symmetric. But if you have that, like I coded this up for you, you can find it here. Then you can say get final composition, and what you get is exactly the same vector here. You can see like 0.577 is this, right? But it would take a much shorter computation time, and especially when you have many species, this thing can take a while. All right. So in the case above, we saw that we started with 7 species and we ended up with 5. Right? Can we say something more general about this process? And the way my laboratory always like jokes about this issue, we call it the problem of the random zoo. And the idea of the random zoo is that you go into a big city that has a big zoo, and then you fence the area of the zoo off. Then you go inside the zoo and you open all the cages of all the animals and just let them free, and then come back after 50 years, right? And the question is, how many species do we have after 50 years? And like the zoo is nice because like these species were not chosen to coexist with each other. They're random to some extent species. There are species that we like to see in a zoo, and therefore like they haven't really established these interactions before, right? Like we will have a lion that is looking at the penguin thinking, what is this thing? They've never seen it before, right? So that's the idea of the random zoo. And with this kind of a code at hand, we can actually simulate the random zoo and start with maybe several observations. So let's say we have 5 species and we repeat this random zoo experiment with these 5 species 2,000 times, right? Just to see how many times do we get all the 5 species, how many times do we get only 1 species and whatnot. And so what we can do is just like for each one of the simulations, build a matrix that is stable, draw random growth rates, find the final composition using this Lemke-Hosson algorithm, and then count how many components of X star are positive. That's the number of coexisting species, right? And then we can take this data and plot it. So this is the result of our simulations, and you actually have code. You can do the simulations with larger number of species or whatever you want. But what I just want to point out is that this is a distribution with what? With a very strong central tendency. The probability of having 2 or 3 species is the same. 1 and 4 is the same. 0 and 5 is the same, which is what? The binomial distribution again. In fact, we can overlay the binomial distribution here, and you can see that if we had chosen like 2 million simulations, we would converge perfectly to the binomial distribution. So this is an interesting result in the sense that a neutral, to some extent, model, right? Like a model where species do not interact with each other, gave us the binomial distribution. A model with random interactions, right? Strong, but random interaction gives us exactly the same result. And this goes back to a discussion we were having in the first class. Right. So how do you go about proving a result like this? That is an interesting question. And I just want to outline this proof that is like something that Carlos did, which I find it very, very beautiful. And so like this proof has two pieces, right? Like one, you have to find what is the size of the number of species that coexist, and then what is the probability that this would be non-invasible. Let's just look at the first part of this. So let's say I want to show you that the probability of having all species coexisting, right? So that would be the 5 here, is exactly 1 over 2 to the n, right? So if, in this case, because the matrix is stable, so all we care about is feasibility. So to have full coexistence, what we need is to have a feasible equilibrium point, which you can write as like the negative product of like the inverse of A times R has to be all positive. Right. So to do this, Carlos concocted this trick that I like a lot. So imagine that you have a certain matrix, which is exactly like the identity matrix. So let me maybe draw it here. So D of K is like a matrix that has 0 everywhere but in the diagonal. And then in the diagonal, it has all ones, besides in position K, where it has -1. Okay, so this is my DK. And now we have to think of what happens when I take this matrix DK. I use it to multiply left and right the matrix of interactions. And I use it also to multiply the vector of growth rate. So what happens when I do this operation is that I flip the kth sign of the equilibrium x star, right? So imagine that I have all positive equilibrium already. I choose one of these numbers to be -1, by say the first one, and then apply this transformation. And what happens is that now x star of the first species is negative, while it was positive before. So I can use this to basically change my parameters to make an equilibrium feasible. Right, let me just show you this in practice. How does it work? So for example, here we build a certain matrix that is stable. We build random growth rates, and then we solve for the equilibrium. And you can see here that the first component is negative, right? So this is not a feasible equilibrium. But now if I choose then DK with like the first element of the diagonal to be negative, let me just clear my drawings, right? Now, what do I have? That when I solve this new system of equations, right, like that is like the modified matrix A and the modified vector of growth rates R, now this equilibrium is positive. Okay? So then we can do this mental experiment of thinking. I have my matrix, and then I multiply this matrix using this left and right multiplication an infinite number of times, right? Like as many times as I want, or let's say n times. So if I choose appropriately where to put these numbers, like, or the order in which I do these operations, then I can always recover a feasible equilibrium, right? So recovering a feasible equilibrium is akin to having chosen the right sequence of matrices to use to multiply this equilibrium, right? In fact, this transformation, because like the eigenvalues of K are the numbers on the diagonal of dk, and its inverse, it's the same as dk, it's a similarity transformation, right? So what it means that when I do this operation, I change the equilibrium, but I do not change the stability of matrix A, right? So if I take my matrix A before the transformation, you can see that this was a stable matrix, right? It has only real eigenvalues that are negative. If I compute the eigenvalues for my modified matrix, right, where I multiply left and right by this matrix D1, I get exactly the same eigenvalues, right? So if the probability of getting a certain equilibrium to be feasible is just like the same as having chosen the right sequence of these matrices decay, then you can see that this has to be 1 over 2 to the n, right? Just because, you know, I have to choose which one should I flip to -1 or 1. And in fact, like you can think that the matrix A and this matrix D1, A, D1 are actually sampled from the same exact ensemble as before, right? So these are still symmetric matrices, they are stable, with the parameters chosen in exactly the same distribution. Right, using the same argument, and you can see like the paper for a full proof, but using the same argument, you can prove that under this parameterization, the probability that these, you know, k species coexist and n minus k's are not able to invade is exactly like the binomial distribution that you can find here. All right, you can complicate this model as much as you want. Like I would not suggest doing that because then the math gets a little difficult. But for example, in the paper we have, what if now my matrix A is fixed and I choose random R's and the matrix does not have a mean of 0, right? Like, or something like that. And what you find in general is the same, that you start with a bunch of species, you end up with a much smaller, typically a number of species, but the proportion is fixed by the parameterization. So if I choose like my R's from a certain distribution, my A from a certain distribution, I have a typical proportion of species that persist, right? So what you would think at that point is that the proportion, if I were to plot initial number of species versus final number of species, what I would find is that there should be some sort of a line, right? Like it's directly proportional to each other. Now, you might think that this random zoo is a bit like of a contrived example, right? Very artificial, but I would like to argue that this type of experiment is actually performed by scientists all the time. And just to prove my point, I just like want to discuss briefly this paper by Leonora Brittlestone and collaborators in like the lab of Otto Cordero at MIT. And what they did in this paper that is referenced here is to sample pitcher plants, right? These are like carnivore plants. So they have this pitcher, and inside the pitcher, there's like this liquid that they use to digest insects, and this has a very rich bacterial community. So what they did is they took the bacterial community from 10 different pitcher plants and then cultured this community in some synthetic medium that they concocted, right? Which is like normal medium, but I think they put some sort of plant extract as well. And what they did is that then they serially cultured this for I think about 2 months, right? So this is somewhat a random zoo, right? We pick a community that would have its own interactions, and now we put it in a different environment where we might think that these species would establish some sort of random relationship with each other. And what would happen then? That like these species could have a positive growth rate in the new environment or not. If they don't, they should go extinct almost immediately. If they do, then they could potentially grow, but because we started with like a little bit of this community and now it's growing in time, like they will feel these interactions stronger and stronger. So eventually like interactions between the species will kick in, and then we should be in the random zoo experiment again. And so I want to like, this is, I'm reporting like the figure 2 of the paper. And so here you can see like on the x-axis is the number of ASV, or imagine of species to some extent, that they found at day 3, right? So once like all the species that had a negative growth rate died. And then on the y-axis, you find the number of species that were there after 2 months. And what you can see is that there is a striking linear relationship between these two quantities, which is exactly what we would expect under some sort of random zoo. All right, now in the let me just go over the chat to see whether there are questions that are important. Oh yes, I forgot to post the lecture notes again, but somebody beat me to this. Thank you very much, Alfonso, for doing that. And then in the random zoo model are also the interaction coefficient chosen randomly. Yes, they're sampled randomly from a normal distribution, and you can see in the code above. So they're just like a random symmetric matrix. And then all we're doing is to change the diagonal a little bit to make it stable. Right? So these interactions are sampled from a distribution centered at 0, and it's a symmetric matrix. You find the code here, right? So this says sample them randomly from a normal distribution with mean 0 and variance 1. Martina asks, can we really assume that the interactions among species sampled from a pitcher plants are random? Absolutely not. in fact, like we think that like they already assemble inside the pitcher plant and they are able to some extent coexist if you're able to sample them because of that. What I'm saying is if we take this community and then we put it in a completely different environment, these interactions may change, right? Because these interactions we're thinking are mostly like due to the resource utilization or things like that. So if you change the resources, you're changing the interactions. So just to be sure, it's not abiding by all the rules of the random zoo. It's more of like the random zoo is a very simplistic metaphor of what we're trying to do. And can we give species a functional role? I'm sure you can. I don't think they went into, there were very many species like you can see here. So I don't think they characterize fully all of them, right? But they started with like more than 120 species sometimes. So it's a bit difficult to see what each species is doing, and would require a lot of experiments. All right, but just to say that even like this very theoretical, abstract model of a random zoo can have some sort of, at least helps us forming some sort of null expectation of what we should be doing. And in fact, like I can tell you that the day like Lenora came to Chicago to present this talk, and I jumped on my chair and I was looking at Carlos saying, have you seen that? You know, when she presented, and then I was talking with Otto actually at another like meeting organized by ICTP, and we had a lot of fun thinking about how do these two experiments relate. All right, now I just want to give you a little taste for this one at a time assembly, like this bottom-up assembly. And so let's try to take the same matrices, the same type of parameterization, but then at each step, we introduce a species at low abundance, and we start from blank slate, from an empty community. We compute the new configuration using this Lemke-Holson algorithm, and then we introduce another species, and then we repeat this process a bunch of times, right? So we can see like the assembly of the community in time, right? So this would be code to assemble one step, right? So we have our matrix, where what we assume is that a certain number of species, a certain proportion of like these X would be zeros. Those are the species that are absent. Another proportion would be the ones that are present in the local community. We pick a random species, could be one that is already there or not. And then we introduce it at very low abundance, get the new composition, and then repeat, right? So we start with 10 species. We build a matrix that is stable. We build random coefficients exactly as we did for the random zoo. And then we start with like X being empty. There's no species. And then we just do 40 invasions, right? And then we track these invasions in time and see what happens whenever we add another species, right? So just to say, for example, here, you know, we start with the invading by species one. Species one happens to have a negative growth rate. It dies. Then, you know, we have like species two that can establish, and then, right? And then we introduce now species one. When species two is present, species one can invade. And in fact, like it resists like the invasions by some other species. If we plot this, which I think it's a little simpler, you can see here we're at the blank slate. A species tries to invade, fails. Then we get species two. Then we get 1 and 2. Then at some point, species 9 comes in, kicks out species 1, and now we have 2 and 9. 10 attaches to the community, 8 attaches to the community, 6 comes in but kicks out species 10, etc., etc., etc. And at the end, we arrive with these 1, 2, 4, 5, 6, 7, 8. Right, so this would be like the assembly. If we were to track the number of species, you can see that we go up, but sometimes we also go down, right? Like here we had 8 species at some point, at time step say 30, but at time step 40, we have only 7 species, all right? Contrary to what you can do in the real world, in a computer, you can go back in time and do this experiment again with the same exact parameters. So we now go to a parallel universe where we can assemble the system again. Okay, 1 second. All right, let's roll back history and assemble again. And if we look at this graph, now we have a slightly different picture, right? So we start with species 4, it resists a bunch of invasions, and then finally species 10 comes in, attaches to the community, 3 comes in, etc., etc., etc. We get to 8 species, and then we fall back to 7 species. 1, 2, 4, 5, 6, 7, 8, which is exactly what we had before. Turns out that exactly because of the saturated equilibrium, we can do this experiment as many times as we want. And we will always find that at the end, if we really wait for long enough, right, such that all possible invasions have happened, we would end up always with the 7 species coexisting together. And in fact, these are the species that I would expect if I were to throw all the species together, right? So in this case of these very stable matrices, the assembly is not super interesting, right? Saying, if I wait for long enough, I always go to the same place, okay? And next lecture, we will actually try to have a pictorial representation of this type of phenomenon. This is not the case when the matrix is not symmetric, right? For example, and in fact, I mean, there is like an interesting observation that you can find in the supplement of this paper, that the probability of finding a system whose final composition cannot be assembled one species at a time. So imagine that we could have like some sort of system where we can get one species at a time up to a certain level, but then to reach like another composition, we have to invade with 2 species at a time, 3 species at a time, 4 species at a time. And again, it's something we will discuss in the next lecture. But our feeling, at least for random coefficients, is that even when this matrix is not stable, the probability of finding such a system goes down quite quickly with the number of species. Now, another thing that I just want to raise as the last point of today, just because it's fun and just because it's like something that leads to a lot of misunderstandings in the field, right? So we go in the field, and as Martina was saying before, right, like these communities that we sample are there, right, persisting happily, and as such, I don't think they should be random, whatever random means, right? And in fact, I can find some sort of like ideas in the literature saying they must have been somewhat like selected in a very non-adaptive selection type of way to persist, right? Like that if I start with something, there's this sweep, like that is like this selective process. Like imagine that the bottom in the top-down assembly would be I throw all the species together, dynamics removes all the species that cannot fit in, and now I end up with a final configuration that is different from my initial configuration, and we saw that there's a different number of species. But did like this pruning select for certain patterns of interactions or not, right? That's the question. So let's do this experiment. We start with a much larger sample, right? Say imagine 200 species, random coefficients, and then we get the final composition, and then we contrast our initial matrix A with our pruned matrix, right? Like the A of only the coexisting part of the community. And similarly, we contrast our initial growth rates with the growth rates in the community, right? So now we can ask, are these two matrices similar, right? That is not an easy question like to answer, because what does it mean for two matrices to be similar? One thing that we could think is like, let's look at this matrix as a graph, right? Like imagine that we take all the coefficients. If they're big, we draw an edge between I and j. If they're small, we leave it alone, right? So we form a graph of strong interactions between these species, right? So this would be the spaghetti ball that is the first matrix. So as you can see, it's like a very dense, random looking graph. And then this is like the pruned matrix, right, which will be fewer number of species and fewer connections as such. It looks also quite random. So this is not the greatest way to visualize whether these two matrices are similar. You know, being who I am, I like spectral graph theory or spectral like methods. So what we could do is to take these two matrices and plot the distribution of their eigenvalues. These two matrices are symmetric, so they will only have real eigenvalues, and we can look at what is the shape of the distribution of the eigenvalues. Here we are helped by what? That we know that if we take a random symmetric matrix with like coefficients that are sampled for a certain distribution, you can actually determine analytically what the spectrum should look like, what this distribution of eigenvalues should look like, and in fact this is called Wigner's semicircle law. And so we plot the eigenvalues of the initial matrix, and sure enough, you see they fall in this kind of shape, which is like Wigner semicircle law. And now we can do the same on the matrix that was pruned, right? Like selected by the dynamics. And what we find is exactly the same, right? So these are like two histograms that look exactly like you would if you were to just sample species at random, right? So it seems that this selective pressure imposed by the environment does not really leave much of a signature in the structure of these matrices. Let's look at the growth rate. Now here we expect something to happen. Why? Because if you have a negative growth rate in the environment, you're gonna die, right? If you have a positive growth rate, you might survive, but you only might, right? Because you're interacting with all the other species. And in fact, like the strongest, the positive growth rate, the more likely to some extent you will be to survive. And in fact, you can see that this is like the red is the initial distribution of growth rates. And in blue, we have the final distribution of growth rates. And you can see that this has been shifted by the dynamics. I have no way right now to calculate the distribution, the expected distribution at the end. So if somebody is really good with this type of problems, go ahead. This is a fun problem to work with because no one has the answer. All right. And so this is like, well, we took random species, did like our dynamics. We end up with basically no like a random growth rate, but from a different distribution, and the interactions look exactly as they did before. Now let's try to do a different exploration that maybe it's closer to what happens like in nature, right? Like instead of say, I just randomly pick species from the whole world and throw them all together, I could say there is one species, and then the new species that comes in is actually a modification of the previous species, right? There's some sort of mutation and reinvasion, like, I don't know, two populations like split geographically, they evolve independently, and now they try to reinvade, right? So this would be a different type of assembly. And this would boil down to what? Like the parameters of like the kth species and the k+1 species should be somewhat similar, right? So what I'm doing here is like I take some matrix, I start with just one species that interacts with itself with the coefficient of -1. That's the only coefficient in the matrix. And now I start building this matrix by saying species 2 will have very strong interactions with species 1 and with itself, maybe a little weaker with species 1 than they do with itself. And then I build this matrix, and then I make it stable exactly as I did before, right? So this matrix, now you can see that this is not a random matrix in the sense that there is this very strong, diagonal, and then you're interacting very strongly with the species around you, right? And in fact, this you can actually see if you do, again, as we did before, plot this graph, right? Like you plot a graph of interactions, and you can see that there's some sort of like gradient, right? Like species that were maybe early on in history, species that were very late in history, and then everything in between. So this kind of axis maps into basically like the time, right? And then when I take this and I prune it, of course, now I get many fewer species, right? Why is that? Because they interact very, very strongly with each other, and they're very similar with each other. But you can see that the structure is basically the same, right? And so again, like we look at the eigenvalues, these should follow some other distribution, which I'm not going to talk about. But then you plot the before pruning and after pruning, and again, you find basically the same distribution. So what this means is, if we throw in like all the species, and these species have a matrix of interaction that is structured, what we get out is a structured matrix of interaction. If what we put in is a random matrix of interactions, what we get out is a random matrix of interactions, which then tells us what? That when we go in a system, and we're saying, oh, look, these interactions have a structure, this structure must be there for some reason. The reason might be that this structure was already present in the pool, right? Like this structure is due to this way with which like we explore the space of possible species rather than having some sort of adaptive, right? And this actually is in connection with like the work of Gold and Lewontin in 1979, famous paper on the spandrels of San Marco. And in fact, even more, and that's what inspired me to write this paper, the work by Ricard Soler and Sergi Valverde in 2006, where they were making this argument for network spandrels, right? So this is like a paper that we published with Dan Maynard and Carlos Servan in 2018. If you want to read it, I think it's a lot of fun. So that's what I wanted to say for today. So right now I'm just gonna go in the chat and see whether I can find some interesting questions. And Antonio, if you have other questions, please go ahead.
Thank you, Stefano. Let's open the floor to the audience if they want to raise their hands or type further questions.
Yeah, there is a technical question by Kiseokli saying, what happens when we subtract this diagonal from the matrix to make it stable? To the eigenvalues, not very much, right? All the eigenvalues will be just shifted of a certain quantity. These will, however, influence like the probability of finding like a feasible random growth rates, sure, and it's in a non-trivial way, so that again, we don't really have a full control over this problem, but certainly we'll do something. And the saturated equilibrium, as I said, is like this solution where we go to where a certain number of species are present, the other ones are absent, cannot invade.
We have a question from the YouTube chat. I can read it out for you. Is there a way to distinguish if two different collections of experimental observations, only equilibrium abundances, were assembled using different metrics and symbols?
The answer, unfortunately, is not that I know of, right? Maybe you can look into that. What I'm just saying is, like, let's not take for granted that if I sample a certain community, I measure all the interaction strengths, which there are methods to do, like Don is actually perfect, and I find that this is a highly structured matrix, then I'm saying this must be the product of some sort of pressure that led to a very interesting configuration, right? Like what I'm saying is that we should always allow for the hypothesis that this structure is just a byproduct of the way in which we chose the species to put in the first place, right? Because the ecosystems are not assembled by taking all the 8.9 million species on Earth and just throwing them all in the same environment and see what happens, right? Like we can only invade with species that are geographically close, evolutionarily like not too distant, etc., etc. Yes, so is the symmetry in A assuming that competition mutualism are the only relevant interactions? To some extent, right? Like you could do the same like with only negative numbers, say only competition, then you add a constant, it complicates the calculation slightly, it's not hugely important. If it's not symmetric, the problem there is not really the non-symmetry of this thing, right? You could do this for a non-symmetric matrix that abides by the same rule that only gives me equilibrium point dynamics. So it would that like to be a very special non-symmetric matrix. The problem is that when A is not symmetric, I cannot even speak of this problem to some extent because I could have like chaotic attractors, cycles, and whatnot, which makes it like very difficult. That was problem number 3 that I tried to remove. Right, so that is why I'm not talking a lot about non-symmetric. In the paper with Carlos, we actually look at non-symmetric matrices that again abide by these 3 rules that I set out. It's a slightly more involved formulation, so I didn't use it for the lectures. Yes, so Adita asks, what happens in bottom-up assembly when the new species are mutants of existing species, right, with very correlated interactions? What will happen is that they're very similar. I would expect that it's very unlikely that they both will eventually be in the mix at the end of the interaction, at the end of the dynamics. And so what happens, in fact, like if you look in the paper on the network expanders, is that it takes a lot longer to build a system of 100 species. Maybe in the random case you would need, I don't know, 300 invasions. There you would need maybe 15,000 invasions to get to 100 species coexisting. And there are some raised ends. Thank you, Flavia. But I don't know if I can see that.
Ankit has a reason, but I think...
Yeah, hi. Can I ask my question?
Yes, please.
Okay, yeah, so like you have this result about getting a binomial distribution for the number of surviving species, where you had a fixed mean of 0 and some random interactions for the off-diagonal. So like, is the variance of the interaction matrix fixed in that case, or like, how does it work?
Yeah, like you can show that this is going to be the same if you choose any distribution for the growth rate and the interactions that is centered at 0. It doesn't matter the values.
Okay, because I've seen in some cases where like the number of surviving species, like it changes with the variance, and
Oh, yeah.
like you also lose stability after a certain point, or is it because of the symmetric nature of the matrix?
Yeah, I actually agree 100%. I wrote several papers on the effect of the variance and the correlation in these matrices and how does it affect stability. In this case, it's like a little special because these things are centered at 0.
Okay.
Right, and I think that is, and also like, yes, these are symmetric, stable matrices, so we don't have to worry about stability to some extent, because the problem of the variance is really like that it makes this distribution of eigenvalues, say, like more spread.
Thanks.
Jordi also has a raised hand.
Yeah, can you hear me?
Yes.
Yeah, first, thanks for this awesome set of lectures, Stefano. And my question is maybe a little bit more speculative, and it's regarding the spandrels idea.
Mm-hmm.
Do you think that there would be instances in maybe some other ecological ensembles, for example, in microecology, where the precursor set of structures are not so stable, so to speak, because maybe evolution is happening in situ, or at the same time as you could see the assembly going on, and therefore we will not be able to see these stable structures, or maybe that's not a very good assessment. Thank you.
Yes, like, yeah, like, of course, these are like cartoons, right, of what's going on. Of course, species are adapting to each other, co-evolving, or like trying to find like solutions to this problem of surviving, you know, in this environment. So that becomes a very complicated, I would say, process. But it's interesting here is really that this is going to be true for species, right, invading each other. It's also going to be true for, like, say, gene regulation network. And in fact, like, if you look at this paper from Soler and Valverde, they're thinking more about gene regulation network and saying, do we observe like gene regulation networks that have a certain pattern of motifs because these motifs are useful to some extent, right? They provide some sort of structure like that is, I don't know, stable or like robust or something like that. Or is it just because like the process of gene duplication is very much like this second example that we were saying, right? That you duplicate a gene, now you have 2 copies of the gene, you can change one basically freely because you still have the other copy, but their function would be somewhat correlated, right? Like, so that would give you only a certain way of exploring the space, which would be reflected then in the structure of this network.
Thank you. Yeah.
So if there are other raised hands, otherwise we have some questions in the chat. Oh, but it's also 9:45, so I don't know if I should stop. But Antonio, what do you say?
If you wish to answer a few more questions, I think that's not a problem.
Sure. So somebody asked, what would be the biological meaning of having identical, similar eigenvalue distribution? Does this provide for similar stability? Yes. Like you can imagine that like any class of, say, random matrices will produce, if it's large enough, like about the same like distribution of eigenvalues, and they will have basically the same properties. Yes. And then, could we see the top-down assembly dynamics as an approximation for any strong disturbance in which new niches are created? That is an interesting question. Like, I'm thinking more of like, you know, some sort of like event in which, you know, we're thinking of invasion always as like one little thing coming in, but many cases, you know, maybe you get like a whole bunch of species because it's brought down like by these, I don't know, animal brings all these other parasites and other things with them. Yeah, but when new niches are created, I'm not sure what exactly it means. So that I would have to think about. Sorry, I cannot respond on. What is the definition of a structured matrix? That is an excellent question. No one really has the answer, right? All the matrices have some structure to some extent. What I'm saying is like that if I draw, for example, this graph, right? This graph does not look like a spaghetti ball. It looks like some class of graph. These, I would say, it has very structured matrix. While if I look at something, it looks really like a random, de Cherney say random graph, then this, I would argue, it has basically no structure. Of course, it has some structure, but it's not like prominent. All right. Anyway, if you have other questions, we have another session. So think of your questions before. If you want to email me like with some questions, I'm happy to answer. And that said, I would just say have a good weekend, and I'll see you on Monday.
Thank you, Stefano. Have a nice
Bye.
week. You as well.
Thank you.
Bye, everybody.
Bye-bye.
Thank you very much.
Thank you, sir.
Thank you, sir. Have a nice weekend.
Thank you. Bye-bye.